Hi, honors biology. We're going to talk about photosynthesis, the overview, and basically there are two phases to photosynthesis, and that's the light independent reactions and the light dependent reactions. Of course, the light dependent reactions occur first, and then our light independent reactions. And basically, we're going to focus is what do you need, where is it happening, and what do you get at the end. Now, as you remember, ATP is the currency of energy in living things, and in cellular respiration, we discussed how ATP is harnessed from glucose, and then how that energy is then used by ATP. And in photosynthesis, we're going to talk about how that light energy is stored in organic molecules to make ATP. And as you can see, all living organisms use ATP to do cellular work, such as transporting proteins, mechanical work like your muscles, or conducting chemical reactions. And that energy flows through the ecosystems, and the two key reactions are photosynthesis and cellular respiration. And our key organisms involved in photosynthesis is our plants, our photosynthetic protists, single-celled or multi-celled algae, or cyanobacteria. They all do photosynthesis. And photosynthesis, for example, in plants occur in the leaf. The leaf is the organ of photosynthesis, and the key organelle where photosynthesis occurs is the chloroplast. Now again, think of chloroplast as two double bags, where the first bag is your outer membrane, and then the second bag is your inner membrane, and inside the second bag is your empty space, which is the stroma. And a stroma is basically a fluid-filled cavity, and then you have the stack of these green discs, and each disc is called a thylakoid. And a thylakoid is just like a sac, so inside is this fluid, and then chlorophyll, our pigment, is in this thylakoid membrane. The whole stack of these discs are called granum, or grana for plural. Another major player we need to talk about is our electron carrier. So when that light excites an electron, the electron is finally accepted by a molecule called NADP. And when it accepts an electron and hydrogen, it becomes NADPH. And then that energy is used to make our organic molecule. Now basically there are two phases in photosynthesis. The first phase is called light reaction, or light dependent reaction, because this is where we requires light. And it uses water, and the water basically is used to replace those electrons that get excited, and our byproduct is oxygen. And we take that energy and capture it into these two molecules, ATP and NADPH, which is then used in the Calvin cycle. And this is where we require carbon dioxide, where the gas, carbon dioxide, is fixed into our organic molecule by a series of reaction called the Calvin cycle, or the light independent reaction, because this part does not require light. And about every 3 cycles, it'll produce the direct product is a 3 molecule called G3P. So 6 of these cycles will produce 1 sugar molecule. So basically, the important thing you need to focus about photosynthesis is basically where is it happening, what are the reactant, what do you need, and what do you get at the end. So here's the equation. It requires 6 molecules of carbon dioxide gas. That's where the carbon comes from in our organic molecule. 12 water molecules. It requires that light energy, and we make our sugar molecule, plus our waste product, 6 oxygens and 6 waters. Now you'll probably see the equation without this water molecule, because when you balance it, this cancels out, and then you have 6. But I wrote the long equation so you know that you also make new water. Now basically there are 2 phases. The first phase is called the light dependent reaction, and this happens in the thylakoid disc. What do you need? You need water, ADP, the precursor to ATP, NADP, our electron carrier. And at the end, we make ATP, NADPH, and oxygen, our waste product. And this is what goes out of the plant. And our second phase is the Calvin cycle, or the light-independent reaction. This happens in the stroma. And the stroma, again, is the cavity, empty space in the chloroplast. And here, it uses the energy that was captured, ATP and NADP. It uses that and carbon dioxide to make our organic molecule. The direct product of Calvin cycle is a molecule called G3P, which is a 3-carbon molecule. And basically, 2 of these would make 1 glucose molecule. So if you look at, with the advent of isotopes, we were able to trace where the atoms come from in our products. So here's our sugar molecule. So that carbon comes from that carbon dioxide gas. The hydrogen comes from the water, and the oxygen in the glucose comes from the carbon dioxide. And also some of the oxygen from the carbon dioxide makes new water. And then the hydrogen, the oxygen gas release is coming from the water. Now, the first phase of light reactions are happening in the thylakoid disc. And the thylakoid disc is basically a membrane, and that's where our chlorophyll is. And this is where we find our electron transport chain. And you heard the word electron transport chain in cellular respiration. So it's sort of like that too. It's in the membrane. This is where the electrons will be passed from one protein to another, and finally will be accepted by an electron carrier, our NADPH. And where these electrons are coming from is the light energy exciting these electrons, and these electrons are accepted through the electron transport chain and transported. Now, while that is happening, just like in electron transport chain and cellular respiration, we're also going to be creating a hydrogen gradient. Now, there's 2 systems, photosystem 2 and photosystem 1, and basically they're chlorophyll molecules where it happens simultaneously, but we'll discuss it separately. And then, just like in cellular respiration, there's also something called ATP synthase that will produce ATP. So looking at the whole thylakoid disc here, here's our electron transport chain, and here's our photosystem 2, photosystem 1. And this is where our light is going to strike our chlorophyll. The chlorophyll will absorb that light energy and excite those electrons. Those excited electrons are then accepted by an electron acceptor in our electron transport chain and passed to one protein to another. And while this is happening, again, you have energy, and that energy is used to pump hydrogen into the thylakoid space. So we're creating a hydrogen gradient here in the thylakoid space, which later on, by chemoosmosis, just like in cellular respiration, is going to be used to go through the ATP synthase to make ATP. And then the light will hit photosystem 1, and this time the electrons are transferred and finally accepted by the electron carrier NADP plus to make NADPH, and which will then go to the Calvin cycle. So 2 things are happening at the electron transport chain. We're creating a hydrogen gradient in the thylakoid space, which will be used by chemoosmosis to make ATP. And at the photosystem 1, we're grabbing those electrons by the electron carrier NADPH, which both of these 2 products are going to be used to make our organic molecule. And just to get a kind of sense where this is happening, again, our electron transport chain is on the thylakoid membrane. Our hydrogen gradient is made in the thylakoid space here, and then the products are released into the stroma. So here's a flow chart that's going to help you kind of remember all this. So remember, in our light-dependent reaction, our light, or you could consider it like photons, sometimes also referred to as photons, hits our chlorophyll. So first hits photosystem 2, all right, and here the electrons get excited, and those excited electrons will get accepted by an primary electron acceptor. and we'll call it P for short. Now the reason I have this arrow rising up here, because when electrons get excited, to show that it increases in energy. And to replace those electrons, we have our water molecule, and our water molecule splits by photolysis, and splits into the hydrogen and oxygen, and those electrons are again replaced by this water molecule, and the oxygen is our waste product. Now, as these electrons are being passed down at the electron transport chain, we are creating a gradient, remember, a hydrogen gradient to make ATP, and later on by chemoosmosis, ATP will be created. Now, then light hits photosystem 1, and again, our electron is excited, accepted by electron acceptor, and an electron is passed down the electron transport chain, and this time NADP is going to accept that electron and be reduced to NADPH, which will then later on be used in the Calvin cycle. Now, what happens is the cell needs more ATP, so this part will happen more often than this part. Now, Melvin Kelvin he's the one who figured out the chemical pathways for the Calvin cycle, or we also know it as light independent reactions. You don't have to remember all these reactions, but basically understand that what you are using is 3 carbon dioxide molecules. We're using the ATP that we made in our light reactions, and our electrons that was picked up by NADPH. And the key thing here is we have this key enzyme called rubisco. So rubisco is considered the most abundant enzyme and that makes food. And this takes this 5 carbon molecule and catalyzes the carbon dioxide to make 2 3 carbon molecules. And this will go through a series of reactions to regenerate this 5 carbon molecule, and after 3 cycles, directly it will produce 1 molecule of G3P. And that G3P molecule, 2 of those will make glucose or other sugar or protein or lipids. So basically what you need to know in the Calvin cycle, it's happening in the stroma. It uses carbon dioxide, and this is what we say, that the carbon dioxide is fixed, or we call this carbon fixation, where we're taking the carbon dioxide in gas form and fixing it into a molecule. And this carbon dioxide molecule uses the NADPH. Those electrons are used, and the energy is used, the ATP that was harnessed from light, to make our glucose molecule. So basically after 6 cycles, it'll produce our glucose molecules. Or you could remember 3 cycles directly produces our G3P molecule, which is basically a 3 carbon molecule. So this is a good flowchart for you guys to review for photosynthesis. Remember, basically there's 2 phases. So here we have our light reactions, and here's where we have the Calvin cycle. So basically our light, and we need water, and we produce a waste product, oxygen, and the key products that we produce from a light reaction is ATP and NADPH, right? And our precursors to that was NADP and ADP, which are recycled after they are used in Calvin cycle. So the Calvin cycle occurs in the stroma here, and this is where we use CO2, and at the end we'll get glucose.